Hello everyone, I am Saurav and I welcome you back to the Digital Board series by Orchids Biosciences. This is the second video for How Do Organisms Reproduce. If you haven't watched the previous videos yet, I have provided its link in the description below, so please do look for it. In this video, we will discuss the asexual mode of reproduction, in which we will discuss the following topics. We will start with fission and its types, which include the binary and multiple fission. Then we will discuss fragmentation, regeneration and budding. After that, we will discuss the vegetative propagation, both the natural and artificial methods. Then we will discuss the spore formation in rhizopus and finally the process of tissue culture. So let's start with asexual mode of reproduction. In the previous video, we have already discussed what is asexual reproduction, in which we have seen that asexual reproduction is a mode of reproduction in which individuals are produced from a single parent. And the individual which is produced will be identical or exactly same to their parent. And this is a very important or useful means of rapid multiplication and is adopted by the lower organisms. These are the key points about asexual reproduction, which we have already discussed in the previous video. In this video, we will discuss the various methods of asexual reproduction, among which the first one is fission. The literal meaning of the term fission means splitting or division. For unicellular organisms, simply the cell division or fission leads to the creation of new individuals. And based on how many cells or how many daughter cells are formed, the fission can be classified into binary and multiple fission. In binary fission, 2 daughter cells are formed, whereas in multiple fission, more than 2 daughter cells or many daughter cells are formed. So fission is a process of simply splitting up of the cell into 2 or more daughter cells. If we see the reproduction in amoeba, it is binary fission because the parent cell here will divide into 2 daughter cells. And let us see how this process occurs. First, the nucleus of the parent cell will divide and form 2 daughter nuclei. Along with the formation of 2 daughter nuclei, each nuclei will be surrounded by cytoplasm, and finally a constriction occurs, due to which the parent cell will divide into 2 daughter cells. So this is how the fission or binary fission occurs in case of amoeba. Not only amoeba, various other single-celled organisms also use binary fission for reproduction. For example, paramecium. The difference between the binary fission in amoeba and paramecium is just that in amoeba, the division can take place in any plane. That is, there is no definite plane in which amoeba divides. Whereas in case of paramecium, the division will take place in the transverse plane. So there is a definite orientation or plane through which the cell divides. So we can see the cell divides only through this plane. And here also the parent cell will divide into 2 daughter cells. So this is about binary fission, where the parent divides into 2 daughter cells. In multiple fission, what happens is the nucleus of the parent cell will undergo many divisions. So one nucleus will divide into multiple nuclei. Finally, each nucleus will be surrounded by cytoplasm. After the nucleus is divided and the cytoplasm is surrounded around each nucleus, during favorable condition, what happens is this parent cell have a membrane, and this membrane will rupture, due to which all the cells or all the daughter cells will be released out. And again, on reaching any suitable or favorable condition, these daughter cells will emerge or develop into the matured ones. So this is what happens in multiple fission. So in binary fission, there will be only 2 daughter cells, whereas in multiple fission, there will be many daughter cells. This can be seen in case of Plasmodium. Plasmodium is a malarial parasite. Moving on to the next one, that is fragmentation. The meaning of the word fragments means pieces, and the meaning of the word fragmentation simply means breaking into fragments or pieces. So with this we can get an idea what fragmentation is. In multicellular organisms which have relatively simple body designs or simple body organization, the body will break up into small fragments. In fragmentation, the body of the organism will break into smaller fragments. Then this fragment will mature to form the adult one again. So in fragmentation, the parents will break into smaller pieces or fragments which will later develop into adult or the matured ones. The example of fragmentation can be seen in case of Spirogyra. After fragmentation, moving on to the next one, that is regeneration. If any individual, for example planaria, is cut into smaller pieces. So this planaria is there, and this planaria is cut into 3 pieces, first, second and third piece. If any individual like planaria is cut or somehow broken up into many pieces, these pieces will then again grow into separate individuals. Here in this diagram we can see that when this planaria is cut into smaller pieces, this head part again grows into new individual, the middle part grows into a new individual, and even the tail part again grows into a fully developed new individual. So here in regeneration, any cut part of an organism has a potential to grow into a fully new individual. This is called as regeneration because here the lost parts are regenerated. This can be seen in case of planaria and hydra. So planaria and hydra can be cut into any number of pieces, and each piece will now grow into a complete organism. The next one which we will study is budding. Hydra can be example for regeneration also as well as example for budding. Hydra when cut into smaller pieces can regenerate, as well as the hydra can reproduce by the process of budding. In budding what happens is one or more outgrowth which are called as buds develop on the body of the organism. These buds will keep on developing on the body of the organism, and later when fully mature will get detached from the parent and become new independent individuals. This is seen in case of hydra and also in case of yeast. In yeast also the cell will reproduce by the process of budding. Moving on to the next one, that is vegetative propagation. Vegetative propagation is a method of reproduction in which the vegetative part of the plant grows into a new plant. The vegetative parts include the parts which are not involved in the sexual reproduction, for example the roots, the stem and the leaves. So the non-reproductive parts called as the vegetative parts of the plant are responsible for producing a new plant, and this process is called as vegetative propagation. It can be natural vegetative propagation or can be performed even artificially. The natural vegetative propagation which we will study are by roots, stems and leaves. Here only one plant is involved, so the new plant which is produced will be genetically identical or exactly same to the parent plant. The various methods of vegetative propagation that happen naturally are by stem, roots and leaves. Potato is a very typical example for vegetative propagation by stem. If you would have noticed, this potato will have many notches on its surface. These notches are called as the eyes of potato. When these eyes or notches of the potato are planted in the soil again, this can give rise to a new potato plant under suitable conditions. So here if we see, without using any kind of reproductive part, just by the vegetative part, potato is a stem. So by using the stem of a plant, we can grow an entirely new plant. So this is how vegetative propagation happens to stem. Coming to the roots, we know that the carrot and the sweet potato are the roots of the plant. Carrot and sweet potatoes are examples for modification of roots which are responsible for the storage of food. When these roots, that is the carrot and the sweet potato, are kept in the soil, these roots can give rise to a new plant. So in this case, we can see how carrot and sweet potato, which are roots of the plant, can give rise to a new plant. These are also not the reproductive parts of the plant, but still it is able to give rise to new plant. Coming to the last one, that is by leaves, the typical example is bryophyllum. The leaves of bryophyllum also have this notches, and at these notches, the buds are formed. When these buds fall, it can originate into a new plant from that place. This is how bryophyllum propagates by vegetative methods of propagation. So these are the natural methods of vegetative propagation. Vegetative propagation can be done artificially as well, and these are called as the artificial methods of vegetative propagation. Artificial propagation includes growing plants by the man-made methods. Some commonly used methods that we will be discussing in this video are cutting, layering, and grafting. So let us imagine a plant like this. This is the shoot system, and there is soil followed by its roots. First one is cutting. In cutting, a part of the stem will be cut, and then this cut end will be planted into the soil. So the part which is cut will be planted into the soil. And after a few days, if we notice, the stem part which is cut from the plant will start growing roots, and finally this will develop into a fully grown plant. So in cutting, we cut a part of the plant, or we cut a stem of the plant, which is then kept in some suitable or appropriate condition like proper soil, it is given proper water and nourishment, and finally this small part which was cut will give rise to an entirely new plant. Cutting can be seen in case of rose and China rose. Moving on to the next one, that is layering. In layering, what happens is one of the stem will be made to bend and touch the soil. So the stem which is made to bend from the plant and touch the soil will start growing roots after some days. And finally we see the part which was bent will later grow into a completely new fully grown plant. So in layering, we do not cut the stem of the plant, whereas we bend the stem of the plant to touch the soil, so that from this part which touches the soil, roots can come out, and this part can completely grow into a fully grown plant. In grafting, there are two plants which are involved. This method involves cutting a twig or a part of the plant from one plant, and then it is joined with another rooted plant. So from one plant, a part of the plant is taken and then connected to another plant, and these two parts are joined by bandaging them. So here we can see that in grafting, two plants are joined together to grow as one. It involves cutting a twig or a part of the plant from one plant and joining it to another rooted plant, and finally bandaging them. So the new plants will have properties of both the plants. So in artificial methods of reproduction, we have seen cutting, in which a stem or a part of the plant is cut and placed in the soil to grow into a new plant, whereas in layering, a stem of the plant will be bent so that it touches the soil and later it grows into a new plant. Whereas in grafting, two plants are used in which from one plant a part is cut and joined with the another part, so that these two parts can join together and grow into a plant which will have properties from both the plants. There are various advantages of vegetative propagation. So let us discuss few advantages. First one is it is very easy as well as a very quick method of propagation of plants. The second one is the qualities of a plant can be maintained. Because it is asexual mode of reproduction, that is why qualities which are available in the parent plant can be maintained in the offspring as well. The plants which are raised by the vegetative propagation can bear fruits and flowers faster, or we can say earlier, as compared to those which are produced from seeds. And if a plant has lost the capacity to produce seeds, these type of plants also can be grown by vegetative propagation. So it makes possible propagation of such plants which have lost the capacity to produce seeds. But these have certain disadvantages as well, because it is an asexual mode of reproduction. The qualities are maintained, but this only should give us an idea of the disadvantage, that there cannot be any kind of variations, and since there are no variations, there can't be no evolution also. Since the same parent characters are maintained in the next generation, there is no variation, and since there is no variation, there will be no evolution. So this comes under the disadvantages of vegetative propagation. Next one is spore formation. Spore formation can be clearly seen in case of fungus, for example, rhizopus. Rhizopus is also commonly called as the bread mold, which we can see on the surface of bread when kept for longer time. We might see some thread-like structures which grow on the surface of bread, and when we zoom into this thread-like structures, we can see something like this. The thread-like structure which we see is actually this part and is called as hyphae. When we observe it under a microscope, we can see some bulb-like structure along with it, and these bulb-like structures are called as sporangium. Spore formation is a method of developing new individuals by forming reproductive structures called as spores, which are produced by this bulb-like structure called as sporangium. So this rhizopus will be having this bulb-like structures sporangium, inside which the minute spores are produced. These spores are very minute unicellular asexual reproductive bodies which are present inside the sporangia. When this spore mature, what happens is this sporangium will burst to release the spores outside. So when these spores are released from the sporangium, the spores will fall on some suitable substratum and will start to germinate again. So we can see that in rhizopus, the thread-like structures that develop on the bread are the hyphae, but these hyphae are not the reproductive part. On the other hand, this blob-like or the balloon-like structures are called as a sporangia. These sporangia have lot of spores which are present in them, and when the sporangium are filled with the spores, they will release out the spores, which on reaching a suitable substratum or which when reaching a suitable condition will start to germinate again. So this is how the rhizopus, commonly called as a bread mold, will reproduce. Finally, let's talk about the process of tissue culture. It is a method of cultivation of plants where new plants are grown by removing tissue or separating the cells from the growing tip of the plant. So in tissue culture, the growing tip of the plant is very essential. From the growing tip of this plant, a new plant is developed. First, what they do is, the cells which are present in the tip of this plant are taken and then kept in an artificial medium. And this artificial medium contains the nutrients as well as the hormone auxins. We've already studied about the importance of auxin in plant growth and development in the chapter Control and Coordination. So here the cells which are present in the tip of the plant will be taken and then kept into an artificial medium which contains the nutrients and oxygens. Here the cells which are taken from the tip of the plant will divide rapidly to form a group of cells which are now called as callus. This callus is now transferred into another medium which contains the growth hormones. This callus is now placed into the growth hormone. Here the callus will further grow and form into small plantlets. So after keeping in this growth medium, this callus will further differentiate and develop to form a small plantlet. After the plantlet is formed, this plantlet is taken out of the medium and placed in the soil. And this have the potential to grow again into a completely new matured plant. So if we try to sum up what we do in tissue culture, first we take the cells which are present in the tip of the plant, then the cells are placed in a medium which contains nutrients and oxygens. Here the cells which are taken from the tip of the plant will continuously divide to form a group of cells which are called as callus. Then this callus will be taken placed in another medium which is containing the growth hormone. Then this callus will soon develop into a plantlet, and when this plantlet is developed, this plantlet will be taken and planted in the soil, after which this plantlet will grow into a completely matured plant. So this is how, from using certain tissues from the tip of the plant, we are able to culture an entire new plant, and hence this is called as tissue culture. So let us try to understand the importance or advantages of tissue culture. First of all, it is a very fast growing technique. Second, the plants which are produced using this technique of tissue culture will be disease resistant. So this is one of the most important feature, and many plants can be developed from a single plant or a single parent. So these are the advantages. First, it is very fast growing technique. Second, the plants which are grown can be disease resistant. And finally, many plants can be developed from a single parent. With this, we come to the end of asexual reproduction. Let us quickly revise what we have discussed in this video. We started with what is fission. Fission is a process of reproduction by unicellular organisms where the parent gives rise to two or more daughter cells. If the parent gives rise to two daughter cells, it is binary fission. If many or multiple daughter cells are produced, it is called as multiple fission. After discussing about fission, we discussed about fragmentation. In fragmentation, we saw that in multicellular organisms with relatively simple body organizations, the body simply breaks up into smaller pieces which are called as fragments, and these fragments will grow into new individuals. After discussing about fragmentation, we discussed the process of regeneration. We saw the regeneration in case of planaria, where if an individual is somehow cut or broken into many pieces, these pieces will be growing into the new individuals. So simply by cutting or breaking up into many pieces, the individuals were able to grow into new separate individuals. After regeneration, we discussed the process of budding, in which a small outgrowth called bud were growing on the parent, and finally after growing to a specific or certain stage, this bud is separated from the parent and then it grows into a complete individual. We saw that budding can be seen in case of hydra and unicellular organism like yeast. Next one that we discussed was vegetative propagation. In vegetative propagation, we saw how by using the vegetative parts of plant, a new plant can be grown. We saw vegetative propagation by natural methods, which were by stem. We saw the example of potato. Then we discussed about the roots, that is, we saw the example of carrot and sweet potato, and finally leaves, that are the leaves of bryophyllum. Then we also discussed the artificial methods of vegetative propagation, which were by cutting, layering, and grafting. After discussing this, we also discussed the advantages of vegetative propagation. After that, we discussed about the spore formation in case of rhizopus or bread mold, where we saw the structures like sporangium, hyphae, and how the sporangium produces spores which on reaching a favorable conditions were able to germinate. Finally, we discussed the process of tissue culture step by step, in which the tissues from the tip part of the plant were taken and then kept in auxins and various other important nutrients, after which it developed into a callus. Then this callus was placed in the growth hormones. Then this callus, after placing in the growth hormone, was able to develop into a plantlet, and finally this plantlet was taken to the soil where it grew into a complete plant. So this is all that we have discussed in asexual mode of reproduction in this video. Let us now quickly take up some questions from the previous years. The first question is split up into two parts. The first part is asking us, budding, fragmentation and regeneration all are considered as an asexual mode of reproduction. Why? This is because in all the three, that is budding, fragmentation and regeneration, only one parent is involved. And the type of reproduction in which only one parent is involved and no fusion of gametes or sex cells happen is asexual reproduction. That is why all the three, budding, fragmentation and regeneration, are considered to be asexual reproduction. That is the first part of the question. The second part of the question is asking us, with the help of a neat diagram, explain the process of regeneration in planaria. For this, we have already seen the diagram. We should draw this diagram and explain it. That regeneration is a process of getting back a full organism, because we can see in all the three cases, the entire complete organism is created. And planaria reproduces by this method, in which if the body of planaria is somehow cut or broken, then each body piece can regenerate into a complete full individual again. So by using this diagram, we can explain the process of regeneration, in which broken or cut pieces are able to regenerate into a complete new individual or organism. Moving on to the next question. The next question is asking us to describe reproduction by spores in rhizopus, that is the bread mold. That we can use this diagram, and then we can explain that the thread-like structure which we can see on the bread is, and when we keep it under microscope and see, we can see some bulb or a blob on a stick-like structure which is called as sporangia. And this sporangia has spores present in it, and when this sporangia is completely filled, and when this sporangia is completely filled, they will be able to release the spores, which on reaching a suitable substratum will be able to germinate again. So by using this diagram, we can explain the process of reproduction by spores in rhizopus. Next question is, what is vegetative propagation, and state two advantages and two disadvantages of this method. So vegetative propagation is a method of reproduction in which the vegetative parts are able to produce a new plant. And these vegetative parts are nothing but the parts which are not involved in sexual reproduction. For example, roots, stem and leaves. So first we can define what is vegetative propagation. Then they are asking us to write two advantages and disadvantages. Advantages, we have studied so many, we can write any two among them. First one is quick and easy propagation. The second one can be the qualities which are present in the parent generation can be maintained in the next generation also. There won't be any change in the character because it is asexual mode of reproduction. This itself can be called as a disadvantage. Here we can write, since the character from the parent will be same going into the offspring as well, there will be no variations. So if there are no variations, there will be no evolution also. So one of the drawback or disadvantage of vegetative propagation is that we cannot expect any variation in the offspring. Moreover, if the species is having any disease, so the parent generation will be having the disease, and since it is asexual reproduction and the same quality of the parent is going to the offspring also, the disease can be also passed on to the offspring. So the offspring is more prone to diseases. So these are the two advantages and disadvantages of vegetative propagation. With this, we come to the end of this video. In the next video, we will discuss the sexual mode of reproduction. Thank you so much for watching this video. If you like this video, please hit the like button, share it with your friends, and please subscribe to our YouTube channel.